Our bladder model with the removed detrusor muscle is a new experimental approach to studying simultaneously the availability of urothelial mediators in the lamina propria and bladder lumen during bladder filling. This model can provide new insights into the signaling mechanisms between urothelium, suburothelium, and detrusor muscle that regulate bladder excitability during filling.
Begin by placing the intact murine bladder specimen into a dissecting dish under a dissection microscope containing 10 degrees Celsius oxygenated KBS and cleaning out the connective and fat tissues around the bladder. Pin a small portion of the dome of the isolated bladder all the way through the outermost edge of the detrusor muscle, far from the innermost edge of the muscle that faces the suburothelium lamina propria, to a Silgard-covered dissecting dish filled with KBS, and pin the ureters and bladder neck to display the intact bladder preparation. Using fine-tip forceps, gently pull a piece of the serosa at the corner between the ureter and the bladder body and adjust the light of the microscope to increase the transparency and to distinguish the margin of the submucosa underneath the detrusor muscle.
To avoid perforating the preparation wall, be sure to cut away the detrusor muscle only while the lateral edge of the bladder mucosa is clearly visible.
Using fine-tip scissors, begin cutting the bladder wall along the inner surface of the detrusor muscle layer while gently pulling the cut segment away from the preparation.